Hello everyone, this is Medical Gateway, and today we're going to study a very interesting topic, which is genetic regulation of transcription. So, what is transcription? Transcription is the formation of RNA from the DNA. So, how the transcription takes place, we have discussed this earlier. This is a double-stranded DNA. Now, these two strands will be separated, and the single strand of DNA will be used as template for the formation of this RNA strand. Okay, now, so the whole process will start at the promoter region, which we will discuss in this lecture, and this whole process is facilitated by RNA polymerase. Now, what exactly RNA polymerase do here? The RNA polymerase basically adds two high energy phosphate bonds to the RNA nucleotides, causing their activation. Okay, so now let's get back to here. So this is the single strand of the DNA which is going to form the RNA. So this single strand of DNA, there is a specific promoter sequence which is known as the basal promoter, from where the transcription is going to start by the RNA polymerase. So this whole is the RNA polymerase, which is attached to the promoter region. And this promoter region has a specific sequence of bases, which is TATAAA. So this is known as Tata sequence or Tata box. Now, Tata box is the basal promoter sequence where the RNA polymerase is attached and will start the transcription. Now we are going to discuss the genetic regulation. It means that which factors are going to facilitate this transcription process by the RNA polymerase and which are going to inhibit this process. So just proximal to the basal promoter region, there are proximal promoter elements. These proximal promoter elements will facilitate the transcription by the RNA polymerase. Okay, so these, first of all, one factor is the proximal promoter elements, which will facilitate the transcription. Then we have the enhancers. Now, enhancers are also a specific sequence of bases on DNA. Okay, and these enhancers will enhance the proximal promoter elements in their activity of facilitating RNA polymerase in transcription. So first of all, proximal promoter elements are sort of promoting the transcription, and these are facilitated by, or their activity is enhanced by, the enhancers, which is a specific sequence of bases on the DNA. Okay, then we have activators. These activators are basically facilitating the role of enhancers and also the proximal promoter regions. These activators are basically transcription factors. Now, these transcription factors will get attached to enhancers, enhancing their function, and they are also attached to the proximal promoter regions here, facilitating their function. And so basically, this proximal promoter element, this enhancer and activator, these all will help RNA polymerase in the transcription process. Okay, now if you see its structure, this is the DNA strand. This DNA strand winds backwards so that this enhancer region comes close to these proximal promoter elements so that they can easily enhance their functions. Now, besides these enhancers and activators which are facilitating the promoter regions, there are also silencers and repressors. From their name, it is pretty obvious that they will inhibit their function. They will silence the transcription process, or they will repress the transcription process, or they will silence or repress the functions of the promoter elements. They are also placed nearby the enhancer regions. They can negatively affect their functions, or we can say this, they just regulate their function. Okay, then we have insulators. So enhancers and activators are supporting the function of promoters and the RNA polymerase, whereas repressors and silencers are sort of silencing or repressing the functions of the promoter elements. Okay, so then we have insulators. Now, insulators have transcription inhibitors attached to them. What insulators can do? Insulators can insulate the functions of enhancers and silencers so that they both cannot affect the promoter elements. Now, what exactly insulators do? Insulators can wind backwards, and they can come between the enhancer silencers and the promoter regions like this. If this whole chain winds back in this form, now this insulator can come between the enhancer and promoter, so they cannot give their enhancing effect to the promoters, and this comes between the silencer and the promoter, so it insulates the promoter from the effect of the silencers and the repressors. So this is the function of the insulator. So these are the factors which controls by which the genetic regulation of transcription happens. So this is pretty simple. In Guyton, this is written in a very complex matter, so you can just simply revise it in the form that promoter, enhancers, and activators will facilitate RNA polymerase transcription function, whereas repressor and silencer will repress the function of the RNA polymerase, the transcription function, whereas insulator will insulate or will insulate the promoters from the effect of both the enhancers and the silencers. So this is it regarding the genetic regulation. See you in next part.